Human Genome Project Whole Genome Shotgun Sequencing. In case of whole genome shotgun sequencing, whole genome is randomly digested. Plasmid libraries are constructed that consist of 2 kb, 10 kb, as well as 200 kb inserts. So in this case, plasmid libraries, they are prepared that can be of 2 kb, 10 kb, and that can be up to 200 kb. Computer programs, they assemble sequences into chromosomes. There is no need of physical mapping while performing the whole genome shotgun sequencing. So this methodology also reduces the problems of repeat sequences. Here we can see in this diagram that the chromosomes, they have been digested into 3 different types of the libraries: 2 kb plasmid library, 10 kb plasmid library, and 200 kb plasmid library. Same is here in this diagram. We can see that the plasmid libraries are prepared of 2 kb, 10 kb, and 200 kb. So there are 3 different types of the plasmid libraries are produced. In this diagram, we can see that how whole genome shotgun sequencing is performed. DNA is purified. It is digested into fragments. These fragments that can be of 2 kb, 10 kb, and also up to 200 kb. Then these fragments, they are sequenced, and once the DNA is sequenced, it is assembled with the help of sequences and by using the computer software. So the overlaps, they are arranged to get the final structure of the DNA. Once the sequence is shotgun, 10 million fragments, approximately 10 million fragments are produced, which are recompiled with the help of computer softwares. Here we can see that these are the sequence reads. Once the DNA has been sequenced, then the sequence contigs are prepared. With the help of these sequence contigs, scaffolds are formed, and then with the help of these scaffold, ultimately genome map is constructed. So this is the method by which whole genome shotgun sequencing is performed. In this diagram, this is the DNA. DNA is digested into fragments. These fragments, they are sequenced, and ultimately we have the sequence of the genome, which is compiled with the help of computer softwares. SLERA assembler was one of the core competencies that make this task possible. Shotgun fragments were compared against each other, and equivalent sequences greater than 40 base pair long were identified. These 40 base pair matches are statistically impossible to occur by chance. So these matches are then determined to be true or repeat induced. True matches are overlapping sections and are the desired fragments in case of whole genome sequencing. The assembler then searches for overlapping fragments that have a common sequence and not contested elsewhere in the data set. The uncontested data is assembled into unitics containing approximately 30 fragments. These assembled unitics are 99% accurate, and unitics passing this filter are ready for ordering and for forming the genetic maps. By looking, these contigs and orientations become the complete mapped scaffolds were arranged to prepare the chromosome maps or chromosome structures. So this is the methodology that how shotgun sequencing is performed. In case of shotgun sequencing, the whole genome is digested into the libraries. Libraries can be of 2 kb, 10 kb, and it can be up to 200 kb. These libraries, they are sequenced, and with the help of computer software, they are assembled, and ultimately the maps of chromosomes are obtained.